Thank you to the organizers for giving me the platform to present our work. My name is Tsitsi Sithandiwe Maponga. I'll be presenting on our study on how functional and species composition of understory plants varies with mistletoe infection on Vachelia karoo trees in a semi-arid African savanna. So I'll start by briefly introducing semi-arid savannas and explain how trees influence heterogeneity and how recent studies have shown that mistletoes have the capacity to improve understory conditions. I'll then get into what the study aimed to investigate, how it was done, and what we found. Excuse me. So semi-arid savannas are often characterized by isolated trees with open and continuous grasslands. These trees and grasses have an existing relationship which shapes the plant community structure. However, fire, for example, herbivory, termite mounds, and large trees are some of the major influencers of plant community structures. So termite mounds and large trees, for instance, they attract sort of like disproportionately more animals by offering shade and nutrient-rich forage. I'm not sure, am I blocking your view? Okay. So large trees also provide and redistribute nutrients to the soil via direct litter input from leaves and also through stem flow and bark slowing. Trees also indirectly increase nutrients through animals depositing excreta while it's visiting the trees. Trees also vertically redistribute water by bringing it up from the bottom layers to the top layers through hydraulic lift, which increases soil moisture levels of the topsoils. Recently, studies have shown that trees further broaden heterogeneity by lodging hemiparasitic plants or mistletoes within their canopies. So these are the mistletoes in the diagram. So mistletoes have high leaf turnover and their leaves have low resorption efficiency, which further increases the nutrients within the canopy patches. If this is coupled with increased animal visitations to the mistletoes, the quantity and quality of litter within the tree sub-canopies increases. Therefore, parasitized tree canopy patches have more litter and nutrients than uninfected trees and possibly intercanopy spaces. The high nutrient levels and suppression of the dominant host trees further influences the species diversity, plant community structures, and the spatial distribution of the understory plants. So studies on mistletoes have shown that there's a difference in the understory productivity and diversity between the infected and uninfected trees. However, no study has assessed how the extent of mistletoe infection influences species and functional diversity underneath low and high infected trees and their intercanopy spaces. So what did we aim to do? We investigated how understory plants respond to different levels of mistletoe infection, that is high versus low, and between canopy and their adjacent intercanopy patches. So we expected that canopy patches and the intercanopy spaces would have sort of like, different species compositions, and also species composition will vary with mistletoe infection intensity. So the abundance of grasses would also be highest in the intercanopy spaces, whilst forbs and trees would dominate the tree canopy patches. Furthermore, we expected that most plant species would have high affinity to the canopy patches, especially those with high mistletoe infection due to the high nutrient levels. Therefore, we expected that species and functional diversity will be highest below canopies of the high mistletoe infected trees. So this is the map that you're seeing. This is the study was done in Matopos at the Matopos Research Center, which is in Zimbabwe. The area has a mean annual temperature of 18 degrees, a maximum temperature of 29.4, and a minimum temperature of 13 degrees. The rainy season ranges from October to April, and the mean annual rainfall is about 586 millimeters. The area is a typical semi-arid savanna. It is partitioned into paddocks, which are used by goats and cows. So how did we do this? We sampled a plot, a 30-hectare plot, using belt systematic transects of 1 kilometer by 10 meter transects with no gaps in between them. So we searched for high and low infected Vachelia carew trees. And with the high mistletoe infected trees, we regarded them as having more than 15 mistletoes, and more than 70% of the canopy was covered by the mistletoe, by the mistletoes. And then for the low mistletoe infected trees, they had less than 7 mistletoes, and they occupied 10% of the tree canopy. So here I've provided the top-down view of the experimental design for each tree. Squares are showing the positions of the 1 square meter quadrats at the different radii from the tree bole in the 4 cardinal positions. So that was giving us a total of 8 quadrats within the sub-canopy and 8 quadrats in the intercanopy spaces. So we had about 4 microhabitats. So we have high mistletoe infection canopy patches, high mistletoe infection intercanopy spaces, low mistletoe infection canopy patches, and low mistletoe infection intercanopy spaces. Excuse me. Species abundance was quantified using the 8-point Walker scale, and measurements were done from around early February and March 2019 during the rainy season, when the species identification was more straightforward. So most of the species were identified at Matopos Research Station, and then the functional trait data was obtained from existing handbooks, field guides, and online literature databases. So we focused on traits that sort of like show the response of understory plants to soil resources, such as life history, life form, photosynthetic pathways, and nitrogen fixation, and whether the plants were native or exotic. We use biodiversity R to calculate species diversity indices, and then functional diversity was calculated using FD and vegan packages in R. Then Kruskal-Wallis tests were used to compare species and functional diversity indices across the 4 microhabitats, followed by a post hoc test. Then we developed what we call the famous Mistletoe index, which was derived from the Termitophily index, which originally was developed by Mushaka Lusa et al. 2019. So what were our results? We had a total of 82 species of plants from 25 families. This comprised of 29 grasses, 31 forbs, and 22 trees. So grasses in the intercanopy spaces were of average to high grazing value, hence indicating that these spaces were less disturbed by livestock. Then low mistletoe infection microhabitats. So that means both the canopy patch and the intercanopy spaces had high abundance of high grazing value grass species, while its high infection microhabitats had mixed species ranging from low to high grazing value. And then when it came to species affinity, 34% of the species showed a strong affinity to canopy patches, while its intercanopy spaces had 9% species that had a strong affinity to them. Also interestingly, 15% of the species showed a strong affinity towards high infection canopy patches, while its 10% of the species showed a strong affinity to low infection canopy patches. So there was a difference of 5% between the canopy patches. As you can see from the first figure, the number of individuals in each of the 4 microhabitats was not different. However, species richness and diversity varied between the intercanopies. Furthermore, both intercanopies had significantly lower species richness and diversity than their adjacent canopy patches. You can see from the from the red eclipses, that high and low canopy patches had different understory species richness and diversity. But in general, all the 4 microhabitats, across the 4 microhabitats, high mistletoe-infection canopy patches had between 52% and 45% more species richness and diversity, respectively, than the low mistletoe-infection canopy spaces, which had the lowest species diversity and richness. As we move on into the functional diversity indices, high mistletoe-infection canopy patches had about 30 to 59% more functional dispersion, evenness, and functional richness. So that's dispersion, evenness, and richness, but there was no difference in the divergence, as you can see from here. So what were our major findings? Our mistletoe-phily index results show that many species had a higher affinity towards canopy patches compared to intercanopy spaces. And at a microhabitat level, they were mostly attracted to high mistletoe-infection canopy patches. So what does this mean? The proliferation of mistletoe infections on Vachellia carobe trees seems to result in changes in the species composition and functional traits, and it also increases species richness and diversity. So mistletoes further, we can say that mistletoes further enhance the effects that tree canopies have on the spatial heterogeneity of nutrients and vegetation structure and composition. So this suggests that canopy effects are far more important in determining overall vegetation composition and structure, whilst the mistletoe infection effects are additive rather to the already existing spatial heterogeneity in these semi-arid savanna systems. High fertility and the weakening of the mistletoe-infected Vachellia carobe trees could have also increased species diversity by supporting a diverse suite of understory competitors within the canopy patches compared to the intercanopy spaces. Moreover, we've found that mistletoe-infected tree canopy patches have higher concentrations of soil nitrogen, phosphorus, and potassium combined. Thus, tree species and forbs were probably capable of outcompeting the grasses. Furthermore, birds and animals visiting the mistletoe-infected trees could have also transported and deposited seeds from other trees, forbs, and grasses into the canopy patches. And this could have also increased the species diversity that we're seeing. Also grazing within the canopy patches compared to the intercanopy spaces could have produced gaps for subordinate species to grow alongside the dominant ones. And this could have probably increased the coexistence of species, resulting in functional heterogeneity. However, despite this higher animal disturbance within high mistletoe-infection canopy patches, they still had the higher species diversity and a totally different species assemblage compared to even the low mistletoe-infected canopy patches and their intercanopy spaces. The loss of leaves from the tree due to high mistletoe infection could have also increased light incidence, and this could have attracted unique species that are tolerant to semi-shaded canopy patches. So in contrast to the intercanopy spaces, the intercanopy spaces had higher temperatures and lower moisture contents and grazing intensity. So probably these conditions had significantly low abundances of, they probably induced the low abundances of many plant species, which include forbs and trees, and hence the dominance of grasses. And as we know, grasses are better competitors in spaces with lower nutrients and water, and therefore intercanopy spaces were dominated by that one growth form, which is grasses, and which is composed of similar traits, and therefore intercanopies, we find that intercanopies had lower species diversity, functional richness, and functional dissimilarity. Therefore, we can say that intercanopy spaces have got poor microhabitat occupation and resource utilization. So in conclusion, all 4 microhabitats had significantly different species composition, as we try to show it using different colors. And this can be attributed to differences in the availability of resources across the different microhabitats, thus making them suitable habitats for an array of functional diverse species. This high dissimilarity results in increased niche occupancy and complementarity within the savanna community metrics. So overall, the spatial heterogeneity of plants shown by the variations in species and functional diversity across these habitats may suggest that Matopo Research Station can be resilient in the face of perturbations or stochastic events within the semi-arid savannas. Therefore, our results could be used by conservation managers and ecologists as a baseline for modeling the thresholds to which mistletoes can be conserved, given that mistletoes increase environmental and plant heterogeneity in semi-arid ecosystems. So I would like to thank the National Research Foundation of South Africa for the funding to present at this conference. You can read more about this study. It was published in the Journal of Global Ecology and Conservation. And I would like to thank the organizers for organizing this conference. And thank you all for listening.